If a plant shows a symptom which could develop due to deficiency of more than one nutrient, how would you find out experimentally the real deficient element? So students, to understand this question, we'll split the question. Now, plant shows symptoms like the formation of brown spots or yellow spots, or sometimes even the cells in the tissue will die. So these are the symptoms which is observed in a plant that are growing in a soil where nutrients are present in lesser quantity. The nutrients can be magnesium, copper, phosphorus, iron, etc. Now, how would we find out experimentally? Like, is there any experiment to find out what causes these deficiency? Which nutrient causes this deficiency? Now, to understand this concept, so first let us find out certain nutrients with its associated symptoms seen in the plants. Now, the first example is, in a plant, the deficiency of nutrients causes multiple symptoms. We are taking an example of a nitrogen deficiency. So, if a plant does not get nitrogen in the right quantity, it does not show just one symptom. Multiple symptoms are associated with it. For example, chlorosis and delayed flowering. So, chlorosis is the plant leaf do not produce right amount of chlorophyll. So, this will directly affect the rate of photosynthesis. And delayed flowering is, it takes too much time to produce flowers. So, when they don't produce flowers, fruits are not formed. So, nutrients of one deficiency causes multiple symptoms. And there is one more possibility. The symptoms might be same, but the cause for the symptoms would be multiple. So, in a similar way, deficiency of nutrient can cause the same symptoms as that caused by the deficiency of another nutrient. Here in the picture we can look. These black spots, what we are observing at the corner of the leaves, edges of the leaves, is caused due to copper deficiency. And the same kind of symptoms is seen in the last picture as well. But here it is distributed evenly throughout the tissue. And this symptom is caused due to phosphorus. So that means when a plant has copper or phosphorus as a deficiency, the symptoms seen in the plant would be the same. So it would be difficult to find out what exactly is present in lesser quantity. We can take another example. Another example is for necrosis. So necrosis is when the cells or a tissue die. So here we can see there are patches of tissues which are dying, and this is called as necrosis. So necrosis is caused by deficiency of multiple nutrients. For example, calcium, magnesium, copper, potassium. All these nutrients, when are present in lesser quantity, can show necrosis. Even when calcium as a single nutrient, when it is present in lesser quantity, it leads to necrosis. In this way, it is very difficult to identify what exactly is the real deficient nutrient. Now, to understand this, we can perform an experiment. The experiment is very simple. We need to prepare a dilute solution of all possible deficient nutrient. So, take a test tube, prepare the nutrient broth, but one of the nutrient should be missing in that solution. So, for example, if there are 10 nutrients which we are adding to a nutrient medium, and all those 10 elements are required for the plant to grow, we'll prepare a nutrient broth in such a way that in every broth one of the nutrients would be missing, and this will be added to the 10 test tube, and there would be one test tube where all the 10 nutrient medium will be present, and that will be called as control. Now we add seedlings to each of the test tube and then observe what kind of symptoms develops in the plant. Now if you take an example here, so we can consider this as a control. That is control test tube where all the nutrients are added and the plant does not show any symptoms, and this plant will be called as healthy. So here we have used a tomato plant, a healthy tomato plant grown in a nutrient medium which does not have any deficiency or which does not have any nutrient deficiency. And if we observe all those 10 test tubes with one of the nutrient deficiency, we could find certain symptoms. Now here, when the same tomato plant was grown in a magnesium deficient nutrient medium, so these kinds of yellow spots were formed. When it was grown in a copper deficient nutrient medium, so these kinds of symptoms were formed. And similarly, for phosphorus, we can observe this kind of symptoms. So once the plants are grown, we should observe the symptoms seen in every test tubes, make a table, record the symptoms what we are observing, compare it with a control plant, and then come to a conclusion that this symptom is caused due to deficiency of this particular nutrient. So whenever yellow spots are formed, we can say it might be the deficiency of magnesium. So whenever brown patches are formed in the corner, we can say the symptoms is formed due to copper deficiency.